So now it's time to talk about biotechnology, the amazing tools that have come out of DNA research. There is a tutorial if you want to look at it, and it really does go through more details than we're going to go through right now. But I'm going to go through some of the basic biotechnology tools, and then we're going to do some we're going to actually perform some biotechnology procedures in the lab with the um, lab exercises that are associated with this unit. So the first topic to talk about is the restriction enzymes and their discovery. Restriction enzymes are special enzymes made by nearly all cells to cut up DNA. The reason cells make restriction enzymes is because living things are under a constant onslaught of viral attack, and viruses attack by injecting nucleic acid into the cytoplasm of cells. So the basic weaponry to destroy viruses is to produce enzymes that will cut up DNA. So as it turns out, every species of organism makes their own versions of restriction enzymes. Evolutionarily, related organisms tend to make the same restriction enzymes, and unrelated organisms make different ones. And viruses that can successfully infect us, like influenza, have protections against our restriction enzymes, which causes problems. So we use enzymes to cut up DNA in the lab, and then we can actually move DNA from like a marigold species to corn, or from a human to a bacteria. And we do that because we want ultimately one living thing to be able to make proteins that normally are found in another living thing. We would like bacteria, for example, to make human proteins so we could give those proteins to sick people and make them better. Another procedure called gel electrophoresis actually separates pieces of DNA that we've cut up, and the pieces of DNA will separate by their size and their polarity. So the gel electrophoresis procedure separates the DNA into a striping pattern that looks amazingly like a barcode. And you can actually use that barcode to determine where the DNA came from. That's DNA fingerprinting. So there's a lot of tools used to make all this happen. Agar is something that's important to the procedure. This opaque material that's right here in this apparatus is agar. Agar looks like gelatin, but it's not. Gelatin is, this is going to gross you out. Gelatin is actually basically dissolved animal bone. It's collagen. That is the protein matrix inside of bones. The problem with gelatin is that it melts when it gets warm. Agar, on the other hand, is much more heat stable, but agar is made from algae, marine algae. So this stuff in here is not gelatin, which is what we used in the DNA slash, I'm sorry, in the osmosis diffusion lab, where we used pink jello shots in vinegar solutions to watch diffusion take place. This is agar or agarose in here that's more heat stable, and you can actually move DNA through the agar to produce basically barcode striping pattern, and that can be used to identify where the DNA came from. Gel electrophoresis basically runs on electricity, and we'll do this in the lab. And DNA moves from the negative side to the positive side, so that's from the black pole to the red pole. And you see a black wire coming in here. So this must be the negative side of the gel here. This is the positive side of the gel here. So the DNA is going to move from here going that way on that electrophoresis apparatus. The DNA fragments will basically move mostly by their molecular mass. And this kind of technique can be used to, like I said, distinguish DNA. You really can't see the DNA bands with the naked eye in the gel. DNA is, it's not optically active. It doesn't absorb enough light to actually look dark or anything. So instead, Researchers use a compound called ethidium bromide, which sticks to DNA very strongly. We will not use ethidium bromide in our lab because ethidium bromide is a carcinogen. It will bond to DNA in living cells and cause all kinds of problems with replication, transcription, translation, and DNA fragmentation. So we absolutely cannot use it in our cells. So this right here is showing, you're looking at these are called wells. You're looking at wells in an agarose gel. This one has had DNA solution injected into it. This one has had DNA solution injected into it. This one, a researcher is right now injecting a DNA solution into the well. This is called loading the well. You're going to do this in the lab, and you'll use micropipettes to do this. So I'm going to need to scroll a little bit. Well, I should show this little stepwise sequence here of diagrams. This is what an agarose gel looks like from the side. There are wells in the gel because of the use of combs. We talked about those in class. To create a well in a gel, you actually have to drop a comb into a tray, and then you pour molten agarose in. It's really hot. When it's molten, it's got to be heated up to about boiling water temperature. This agarose will melt, meaning it liquefies, and it can be poured at that temperature. But once it cools down to about 55 Celsius, it'll solidify. And when it's solid, you can pull the comb out, and that leaves little holes in the gel. These are micropipettes. We'll talk about those in a minute. They're used to actually inject the DNA into the wells. And when you inject or load the wells and then turn on the electricity, this is the negative side. The DNA fragments that are in your wells will actually separate, and they leave barcodes, or at least single stripes. And then these barcodes can be used to identify where that DNA came from. This is probably best seen in the biotechnology tutorial at the end with DNA profiling. So this FBI researcher is actually holding up a complex DNA fingerprint. There were wells here, 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 all the way down. Underneath each barcode, there was a well. The researchers put DNA samples from a single person into all those wells, and they ran electricity through the gel, and that caused the DNA to separate. This fragment pattern is unique to that person. It's scanned into a computer, and that goes into what is called the CODIS database. The CODIS database is used to identify criminals around the world, actually. Anybody who commits a crime is basically DNA profiled. DNA profiling is probably a better name than DNA fingerprinting, because when you say fingerprinting, people think you're putting ink fingerprints on a piece of paper, and that's not what this is. This is DNA barcode patterns that are actually in a gel. This researcher is holding up what looks like a piece of plastic, but if you let agarose dry out on a surface, it flattens out and becomes literally a biological piece of plastic. So this is actually an agarose gel that's been dried up, so that all the little bands are really, really distinct and obvious. And then, like I said, this can be scanned into a computer to be stored in a CODIS database to see if it matches anybody that's committed crimes. So let's go back to our biotechnology unit and talk about the micropipette itself. Micropipettes, like the one that's pictured here, consist of basic parts. E is the disposable tip. A is the tip eject button, so when you push A down, the tip flies off. C is the volume button, so when you push C down, you're either pushing a liquid out of E or you're going to suck something up into E, depending on whether you're pushing the button before or after you stick the tip down here into a liquid. And then B is the volume you're actually going to dispense, and here this one says 10.0 microliters. And D is called the body of the pipette. That's what you wrap your fingers around in your palm to actually hold onto the pipette. So what we last need to mention is kind of how restriction enzymes work. And for that, I probably should go to the biotechnology tutorial and scroll up to the top. And let's see, let's do that. And near the top, there's a simple animation of how restriction enzymes work. On this one, basically this enzyme wraps itself on the DNA and does not cut straight through, and it leaves what are called sticky ends. This is called a sticky, let's get the drawing tool. This is called a sticky end here and here. They're sticky ends because they're double-stranded, they're sticking out, and if you mix another piece of DNA with this piece that reads TTAA at the end, it'll stick to this, and then you can use ligase to bond it together. That's how researchers bond DNA from, say, a human into a bacterial genome. So in this case, let's go back to the cursor, and let's step through this. So the enzyme is basically cutting the DNA, and it cuts through at a diagonal, leaving those sticky ends, which is so useful in biotechnology research. Now we need to, so these sticky ends, let's erase those drawings. There, this is a sticky end and this is a sticky end, which can be used to link DNA together. And here's what you see in this animation. That's exactly what's happening. So this is a piece of DNA. This is from bacteria with this sticky end. This is human DNA with this sticky end, and they're complementary. When you put them together, the ligase can bond them, and now we've got a human piece of DNA stuck to a bacterial piece of DNA, which can be very useful for making, say, human insulin in a bacterial culture, which is the best way to make insulin for diabetics. In the old days, pig insulin was used for diabetics. Now it's human insulin made by bacteria. Now, let's talk briefly. Oh, you know, we should probably go through a quick animation of gel electrophoresis. So let's look at gel electrophoresis. You're going to actually do this procedure, but here's the apparatus and here's how all this works. Starting with A, this is this wire right here is the negative wire. The negative wire is always on the well side of the electrophoresis box. B is the positive wire. The A wire is black and the B wire is red by convention. So these are plugged in correctly. A is the black lead plug and B is the red lead plug. C, let's see, where is C? Is the micropipette up here that is used to load the wells. D is the actual wells here, which are little holes left in the agarose gel because of the use of a comb. I'm going to go out of order here. I was the comb that was laying in here, and then when the gel solidified, it was pulled out, and that left these holes. And then let's go to F is the power supply that's going to run the DC electricity through the agarose gel. G is the entire, it's called the box that holds the whole apparatus. H is the actual tray that holds the agarose gel. E is the actual gel that is inside the tray, which is inside the box. So that is the apparatus, and here's an animation of how it works. I'll step through this. So you can barely see it here. Let's make it just a little bit bigger. Let's see, there. Okay, let's go to that. Maybe that will just fit. Yes. So what the researcher is going to do is with this micropipette, this researcher is going to suck a little bit of DNA. DNA is almost always dyed, so you can see what you're injecting. So the researcher is going to suck up this some of this DNA, and they're going to inject it in the well, and then they're going to turn on the power, and the DNA is going to move. Okay, so here we go. So the DNA right there, you'll notice in the disposable tip we have a little sample of DNA. This is probably 5 to 10 microliters, and then there's another 5 to 10 microliters left in the little microtube right there. Now, the researcher is going to push this button, and they're going to squirt. You'll notice the tip of the micropipette actually is not stuck down in the well. And the reason for that is if you twitch your hand at all, this tip will cut the agarose gel, and then you've ruined it. So to avoid that, you only put the tip right down at the top, and then you push this button up here, and it'll inject. The DNA is usually injected with a sucrose solution to make it really heavy and dense, and that will cause this to fall down into the well. So here we go. See, it literally injects like that, so the well is filled up. Then the researcher turns on the power here, and you're going to see this little needle here? This is showing power on. And now that the power's on, DNA threads start to move out. But the heavy ones are going to go slow, and the light ones are going to go really fast. And in this way, the DNA races across the gel, and you got to be around. You can't let this go too long, because if you do, all the DNA will go out of the gel and start floating around in the water here, or the salt solution. So you turn it off, and now this is a stripe, this is a stripe, and this is a stripe, and those are equivalent to these stripes. This is looking at the gel from the top, and this is looking at it from the side. So this is actually a trillion pieces of heavy DNA. This is a trillion pieces of lighter DNA. And this is the lightest DNA that's in this lane. This is called a lane that all separated when this well was electrified, and the DNA moved from the negative pole to the positive pole. You'll notice that the striping pattern here is different, and this one is different, and this one's different. So the 4 wells here had different fragments being moved through the gel. So that is how DNA electrophoresis works. Now let's talk about the polymerase chain reaction. PCR, the polymerase chain reaction, is really important to forensics, because there's really never enough DNA left at a crime scene to actually get a DNA profile. So what a researcher has to do is they have to make many copies of the DNA collected at a crime scene, like a trillion. So the way this works is this. Over 20 years ago, researchers discovered a kind of DNA polymerase that's heat stable, and it's called Taq polymerase. Taq polymerase, TAQ, stands for Thermus aquaticus, which is the name of the microbe collected in a hot spring in Yellowstone National Park that actually makes this goofy enzyme. The bacteria actually lives in almost boiling water. It's like 95 degrees Celsius, which would kill you if you jumped in it. They live in it, and their DNA polymerase and all their enzymes are heat stable, meaning they can handle 95 Celsius. So researchers extracted that enzyme, and we now can make that enzyme in other bacteria and use it in DNA research, especially PCR. So if a researcher puts Taq polymerase along with nucleotides and primers into a test tube with a tiny sample of crime scene DNA, and then put that into what is called a cycler, a thermal cycler or PCR machine, you can end up making, in about 160 minutes, you can make about a trillion copies of the crime scene DNA, which is enough to actually do a DNA profile. And the way this works is this, and I can show you an animation in a minute, but the researchers put the DNA samples into the thermal cycler, or also called the PCR machine. The PCR machine is programmed, and every 4 minutes it goes through a heat, cool, warm cycle. It heats the sample up to almost 100 Celsius. That denatures the DNA, and the DNA literally comes apart. Then they cool the forensic sample down to like 50 Celsius, and that allows little primers to attach to the DNA. Then they warm the sample up to about 75 Celsius, and at that temperature, replication occurs. And then they repeat. Heat, cool, warm. Heat, cool, warm. 1 minute, 2 minutes, 1 minute. 1 minute, 2 minutes, 1 minute. It takes about 4 minutes per cycle to complete a PCR cycle. And if you do let's see, 40 cycles, you get an enormous amount of DNA, a trillion copies, and that many copies is sufficient to do a DNA profile. The way I'll show you an animation that actually kind of shows you how this works. Let's go to the cursor and then go to the tutorial, and we need to scroll to the PCR animation. So scrolling down to it here. Okay, it's kind of here. This is it. So I think this will fit good. So we have here crime scene samples of DNA that have been collected, and they're sitting in a test tube. And so floating around inside of the test tube are heat-stable Taq polymerase, primers, and nucleotides. And so here's how this works. Let's go back to cursor so I can step through. Okay, here we go. All right, so you heat up to 95 Celsius. This denatures the DNA. You'll notice the DNA has unzipped. It doesn't actually break up, but it unzips. And that's what a lot of researchers say, well, it breaks up. Well, this is called denaturing, but it doesn't really fragment the DNA. It just separates it into single strands. At that point, then, you cool. By the way, it did say at the very top of the screen here the temperatures. Oops. Let's go here. So at this point, we're at 95 Celsius. Then if we step, now we've cooled it. This is the second part of the cycle. We cooled it to 55. This is the cooling phase. At this point, the primers go in and attach to the single strand of DNA. Then you warm it up to 75, and that's what you see here. And then at 75, the DNA Taq, the little blue things are Taq polymerase. They move down the line and they replicate the DNA to maybe 300-400 nucleotides long, and then they stop. Okay? At that point now, we had started with 2 pieces of DNA, now we got 4. If you heat back to 95, all 4 pieces denature. If you then cool to 55, your primers will attach to all the appropriate parts of the DNA. If you warm to 75, the heat-stable Taq polymerase will attach to the primers and replicate downstream to replicate the DNA. If you run through another cycle, you will end up denaturing all 8 pieces of your DNA. And what will happen is when you warm the DNA, or when you cool it, so when you heat it, you'll denature it. When you cool it, your primers will attach to all appropriate regions. And then when you warm it, you'll replicate your 8 pieces of DNA to 16 pieces of DNA. If you keep doing this for 40 cycles, you'll make 2 to the 40th copies of the DNA, which is sufficient to do a DNA profile. And that is the foundation of forensic biology in crime scene investigation. If we didn't have PCR, there's no way we could have caught people like Dennis Rabbit or the Golden State Killer in California. We did PCR and then electrophoresis. So once you've done the PCR, then you have to do the electrophoresis, which was this animation back there. You have to do this to produce the DNA profile. And it's actually quite complicated. 13 different regions, I think, on the human chromosomes are actually sequenced to do a proper DNA profile. But once that's done, you can actually get a match to a felon that's committed a crime if they have committed the crime and have that DNA sample at the crime scene. Now, there's a couple of other things we need to mention. Gene cloning is a big deal. And right now in biotechnology, there are labs around the world making human proteins or other proteins using plasmid biology. So what we're doing is we are putting human genes into bacterial plasmids, and then we put those plasmids into live bacteria, and we actually let those bacteria make human proteins. So this kind of biology is it's saving lives around the world every day. What researchers are doing then is they're cloning a whole bunch of bacteria that have a human gene in them. The way you maintain the clone is that the clone bacteria have an antibiotic resistance gene along with the human protein gene that you want them to use to make a human protein. The reason for the antibiotic resistance gene is bacteria are really small, and you never know what's growing on a culture dish unless you can throw antibiotics in the culture dish and kill all stray bacteria that may be falling in your culture. So antibiotic resistance is another backbone phenomenon in biotechnology. So the way this procedure is set up is a human gene and a bacterial plasmid are both cut with the same restriction enzyme so that they have complementary sticky ends. You basically mix then the bacterial DNA and the human DNA together and let ligase seal them up. You then put the plasmid into bacteria, that's insertion, and then the plasmid is replicated along with the genome of the bacteria when they reproduce, and you are essentially allowing the bacteria to clone your plasmid. Along the way, the bacteria will read the gene and they will make a human protein, and we can use that protein to save lives or to help people. Human growth hormone, HGH, and insulin are both made this way, along with a lot of antibiotics, human collagen, and all kinds of other stuff. I've already mentioned the antibiotic resistance, because without that, you can't guarantee that your cultures have only the bacteria you want to grow. Now, I really can't get out of biotechnology without at least mentioning CRISPR-Cas9, because CRISPR-Cas9 is going to revolutionize medicine. CRISPR is a technique where researchers are able to literally